Welcome to BioBits and Bytes. Today, we are diving headfirst into a realm where, honestly, the foundational laws of software and the complex architecture of life are just collapsing into one. We're talking about moving computing beyond the rigid, fast, but fragile world of silicon chips and into something radically different. Flexible, self-healing, massively parallel, and just incredibly energy-efficient living computers.
It really does sound like science fiction, doesn't it?
Yeah.
But this is happening right now. We're no longer content just, modeling biology on a computer. The objective has fundamentally shifted. Now it's about engineering life itself to perform sophisticated computation.
So you're saying we're treating the DNA inside a cell as the actual programming language?
Precisely. DNA is the code, and we're using proteins as the functional nanoscale components that actually execute the logic.
And the practical implications are, they're stunning. We're going to talk about systems that are infinitely more adaptable than anything silicon can produce. For instance, we'll get into how a simple cup of coffee can be programmed to trigger a precise drug release inside your body.
Yeah, and how researchers are programming single human cells to perform complicated logic, like an XOR-ND circuit to identify cancer.
Wow.
What's fascinating here is just the sheer potential for statistical robustness and energy efficiency. Think of the enormous server farms required to run today's AI. A single living cell is a computational unit that runs on almost zero power compared to a traditional processor.
And this convergence, it's drawing from four really distinct disciplines, right? We're talking neuroscience, AI, engineering, and biotechnology.
All of them coming together.
Our mission today is to unpack this paradigm shift. We've gathered a stack of detailed sources that give us a critical computational perspective on neuro-AI, on synthetic biology applications in everything from medicine to environmental cleanup, and on a truly counterintuitive but powerful idea called the constrained disorder principle, the idea that biological noise is actually essential for life. Okay, let's unpack this.
So when we define synthetic biology, the core principle is really designed. We're taking the natural components and machinery of life, genes, proteins, metabolic pathways, and re-engineering them, or sometimes assembling entirely new ones to achieve a predefined desired function. We are quite literally engineering the cell's operating system.
Okay, so if DNA is the archival storage, the hardware, let's say, and the resulting proteins are the functional machines, how do we program the process in between?
We program the flow of information, and this process happens at three distinct layers inside the cell. We can control things at the transcriptional level, which is where the DNA code is read and transcribed into messenger RNA or mRNA. Then we can manage the post-transcriptional stages, where that RNA is processed. And finally, the translational level, which is where the ribosome reads the mRNA and translates it into a functional protein.
So you can insert or modify genetic switches at any one of those three steps.
Exactly. You can control any one of them.
And I guess based on what we want the engineered cell to do, say in a plant or in a person, we need different kinds of logical behavior. Our sources categorize these circuits pretty neatly.
They do. They divide the functional outputs into two main categories. The first are what we call irreversible circuits. These function as a kind of memory element. They're designed to retain a memory of an input signal, so they maintain an output state once they're triggered, even if that original signal disappears.
That sounds incredibly useful for persistent problems. Like, if a plant is engineered to detect a specific fungal pathogen that only appears for a short season, the circuit could detect it, flip a switch, and then permanently keep producing an antimicrobial compound for the rest of the plant's life.
That's a perfect example. It's a permanent on or off state. The second category is, naturally, reversible circuits. These are dynamic. They allow the output state to rapidly and fluidly change in response to varying input signals. It's more like a sensor with a continuous feedback loop.
Right, so if we engineer a therapeutic cell to produce insulin, we'd want that to be a reversible circuit. It needs to dynamically scale insulin production up if blood sugar rises, and then scale it back down if it drops.
Precisely. That dynamic control is key for homeostasis. And this is where the advantage of biological computation truly shines. Traditional Boolean logic, it demands perfect ones and zeros. It works in a rigid, deterministic, singular way.
But living cellular computers trade that singular speed for something else, something superior in complexity.
They do. They use DNA for programming and proteins as their functional components, and that offers these immense benefits. Things like statistical robustness, because the system relies on the average behavior of millions of computing units, the cells all at once.
Hmm.
You get extreme parallelism, inherent self-healing, growth potential, and as we said, superior energy efficiency. What's fascinating here is this shift means trading the speed and exactness of silicon for the resilience and adaptability of biological plasticity.
The theory is amazing, but assembling these complex programs in a wet lab, I mean, that's where the rubber meets the road, isn't it? And it's often where researchers hit a wall. Our sources repeatedly point to molecular cloning, constructing those circular plasmids or vectors, as a fundamental and laborious bottleneck. Why is it such a headache?
Molecular cloning is. It's painstaking. You're splicing, cutting, and pasting DNA sequences using enzymes. You're amplifying things with PCR. The challenge is multi-layered. You have to verify every sequence, purify the DNA fragments, and repeat that process over and over to assemble a complex circuit. But historically, the worst part was dealing with conflicting restriction sites.
Okay, what does that mean?
It means that the piece of DNA you wanted to insert might contain the exact same sequence that you needed to use to cut the vector open.
Ah.
So when you add your enzyme, you cut up not just the vector, but your precious insert as well. Your entire assembly strategy just collapses.
So genetic engineering, much like electrical or mechanical engineering, it quickly outgrew the capacity for manual design and assembly. We needed computer-aided design or CAD software to handle this genomic complexity.
Absolutely. The complexity just necessitates CAD tools, the same way they're essential for designing microchips or aircraft. And so platforms like Genome Compiler emerged to meet this demand. They offer an all-in-one environment. Researchers can digitally design complex vectors, simulate different cloning reactions, and track their inventory of genetic parts, all before they even step into the lab.
And to streamline the actual physical process, Genome Compiler integrated a system called SnapFast. How did that technology address the messy problem of non-standardized DNA parts?
SnapFast. It was a brilliant response to the standardization crisis. It introduced a modular approach to cloning. Instead of every lab designing unique parts that might not be compatible, SnapFast established a core universal vector backbone. This backbone accepts standardized, interchangeable pieces of DNA, we call them parts, that can be easily swapped in or out.
So it's like creating Lego bricks for biology, where every piece snaps into the same designated slots.
That's the perfect analogy. They eliminated the conflicting restriction side problem by standardizing the cutting points. And the sheer scale of the available parts is what makes it so powerful. There's a vast library, 26 peptide tags, 9 reporter genes for visual output, over 40 promoters to control expression strength, 10 selectable markers. This library gives the synthetic biologist precise plug-and-play control over the circuit, which radically reduces the need for laborious manual redesign.
So it shifts the researcher's focus from just assembly logistics to pure design logic?
Exactly.
Okay, so even with streamlined assembly, the cell is a hostile, constantly fluctuating environment. We need to tune the performance of these genetic machines. What are the key tuning knobs that synthetic biologists can manipulate to make sure the circuit behaves as programmed?
Right, we have several layers of control, from the very start of the gene expression process to the final end product. First, the most fundamental control is promoter strength. The promoter sequence dictates how efficiently RNA polymerase, that's the enzyme that makes RNA, is recruited to the DNA. If we use a stronger promoter, we get more transcription. A weaker one, we get less.
That handles the transcription. But once the mRNA is made, how do we fine-tune the resulting protein product?
Well, we can control the speed of production via the ribosome binding site strength, or RBS. That influences how quickly the ribosome docks onto the mRNA and starts translation. And on the other end, we can control the lifespan of the protein by adjusting its enzymatic degradation rate. We can essentially program a self-destruct mechanism for it.
And for sort of large-scale changes to the output of the entire circuit, we can manipulate the system's size through gene dosage, right?
Correct. We can move the circuit from being integrated into the host cell's genome, where it might only be 1 or 2 copies per cell, to a high-copy plasmid, where it could be present 50 or even 100 times. This provides a very broad, systematic way to tune the overall expression level.
But it comes at a cost, I'd imagine, a bigger cellular burden.
A huge cost. It can stress the cell out. That's why we also have more subtle ways to manage things. For example, if we need to manage a repressor protein without redesigning it,
How do you do that?
we can use clever molecular decoys. One method is using small RNAs, which we can engineer to bind specifically to the repressor's mRNA and target it for degradation. Another really fascinating approach uses decoy operators. These are extra binding sites we introduce that do nothing. except bind and sequester the repressor proteins. They titrate them away from the actual promoter site, which indirectly boosts the gene expression we want.
It sounds incredibly intricate, but we've established that the cell is a chaotic, constrained environment, constantly demanding resources. This brings us to a major technical hurdle: host interactions. Why are early synthetic circuits so notoriously fragile?
They're fragile because they don't operate in a vacuum. Every genetic circuit relies heavily on the host cell's resources, ribosomes, RNA polymerase, energy in the form of ATP, amino acids. A small, subtle change in the cellular environment, the growth media, the temperature can shift the availability of these resources, and that can lead to circuit failure or unpredictable behavior.
And this inherent dependency gives rise to a critical concept we need to understand: retroactivity.
Retroactivity is the mechanism by which a downstream genetic element can unintentionally influence the performance of an upstream element. It's the backward influence that happens when we connect a new module to an existing circuit, and it essentially destabilizes the original system's performance.
Okay, so if I understand correctly, retroactivity is like installing a huge, resource-hungry new app on an old smartphone. The new app starts hogging the phone's memory and processing power, and that slows down all the other older apps that relied on those same shared resources.
That analogy perfectly captures the essence of resource competition. Let's look at a specific circuit failure. A simple NOT gates function relies on a very precise concentration of a repressor protein being available to bind to its promoter. But if you connect a second output module downstream, that second module also has to consume or titrate that same repressor protein to function.
So the repressor protein is now split between two jobs. And the first job, the NOT gate, no longer has the precise concentration it needs to work correctly, or at least not on time.
Exactly. This titration weakens the upstream NOT gate. It often delays its response to the input signal, causing the synthetic program to fail its timing requirements. So, to fight this, researchers often just increase the expression of that problematic component, the repressor protein, to ensure there's enough to go around.
But wait, doesn't that massive increase in protein expression introduce a dangerous trade-off? If you flood the cell with extra non-essential protein just to make the circuit work, aren't you stressing the cell or potentially making it toxic?
That's a great question. Let's break it down. You've hit on the central dilemma. You might temporarily fix the retroactivity delay, but you introduce new trade-offs, things like increased cellular energy drain, toxicity, unpredictable changes to the host's native pathways. The synthetic biologist is constantly battling the inherent complexity and resource limitations of the living cell. This is why developing standardized design principles for modularity and insulation circuits that are less sensitive to these resource fluctuations is the vital next step to turn this into a reliable engineering discipline.
We started by comparing living cellular computers to rigid silicon. Now we're going to transition to the ultimate biological computer, the brain. When we talk about neuro-AI and organoid intelligence, or OI, we're talking about systems that move beyond single-cell logic and harness the emergent, massive complexity of millions of living neurons.
This really is the most profound convergence we face today. Synthetic biological intelligence, or SBI, is the umbrella term for the field that integrates living neural cultures, especially cerebral organoids, with advanced electronic interfaces. And these organoids are 3D cultures that manage to recapture astonishingly complex structural and functional features of the developing brain.
What does recapturing those features actually mean in a lab dish? Are they just random clumps of neurons?
Not at all. That's the amazing thing. They develop specific structures, including identifiable cortical layering, and they exhibit spontaneous structured network activity that matures over time, and it begins to resemble early developmental patterns that we see in human fetal brains. We're leveraging the innate, massive parallel computation, learning, memory, and adaptive capabilities of these biological neural systems to achieve engineered intelligent behaviors.
It feels like training a super fast calculator for a living, adaptable, albeit slower, learner. What unique advantages does this plasticity offer?
Well, we retain the core biological advantages we already discussed: robustness, self-healing growth, and superior energy efficiency. But now they're amplified by the emergent properties of neural networks. Unlike digital von Neumann architectures based on binary logic, these post-von Neumann systems harness the complex compositional dynamics of billions of neural elements to generate emergent forms of computation. They learn and adapt in ways we still struggle to fully model.
And the goal isn't just to study them, it's to create hybrid systems.
Exactly. The systems are designed to be highly interactive. We're interfacing them with sophisticated electronic sensors and actuators, even physical muscle tissue. We're creating organic machines capable of probing their environment and actively responding. And this controlled environment allows us to study fundamental biological learning and reasoning processes in action. And beyond computation, this gives us unprecedented platforms to model complex neurological diseases like Alzheimer's, Parkinson's, or epilepsy to test rescue strategies that bypass the limitations of animal models.
To program or even understand these living intelligent systems, we need sophisticated theoretical frameworks. One crucial computational approach that guides the design of these intelligent biological agents is neurosymbolic AI. That term suggests blending the best of both worlds.
It is a hybrid strategy. It combines symbolic reasoning, so the use of explicit rules, structured knowledge, and logical inference, which traditional AI excelled at with subsymbolic learning. That's the data-driven pattern recognition we associate with modern deep learning. The practical benefit is huge. You get enhanced accuracy, greater explainability because the system uses rules, and better adaptability because it can integrate new structured knowledge with raw data.
This connects directly to how we define agency in these biological systems. Our sources highlight active inference, or AIF, as the optimal policy framework for these agents. How does AIF conceptualize and guide behavior?
AIF posits that any intelligent agent, biological or artificial, is constantly trying to minimize its expected free energy. Conceptually, this free energy represents the discrepancy between the agent's internal model of the world, its beliefs, and the observations it receives from the external environment. The optimal policy is achieved by selecting actions that are predicted to minimize this surprise in the future.
That seems like a complex calculation. A key insight of AIF is that this drive to act, to minimize future surprise, is mathematically decomposed into two distinct motives, which are crucial for explaining why living systems explore.
They are. The expected free energy is decomposed into the extrinsic risk component and the intrinsic information gain component.
Let's break those down. What is the traditional goal-directed motivation that's captured by extrinsic risk?
The extrinsic risk component captures the classic goal-directed motivation we see in standard reinforcement learning. It's the anticipated surprise or negative consequences of future observations relative to the agent's preferred states. In plain terms, it's the drive to avoid pain, seek reward, and achieve explicit goals set by the environment or its designers.
And the intrinsic information gain component. This is the truly novel part that explains the biological drive to learn.
Precisely. This is the epistemic drive. It measures the expected reduction in uncertainty about hidden states or parameters within the agent's own generative model. It reflects how much a new observation is likely to improve the agent's internal beliefs. The agent is mathematically driven to seek out information that reduces the fuzziness in its understanding of the world.
What do you think? This insight is profound because it establishes exploration not as a learned strategy, but as an inherent mandate of the optimal policy. The agent doesn't explore because it might find a reward. It explores because it must reduce uncertainty about its internal model.
Absolutely. That's a huge contrast with standard RL, where exploration is often a manually tuned parameter, like the epsilon greedy approach. The mathematical structure of AIF forces the agent to constantly update that generative model, guiding both perception and action. For neuro-AI, this is the blueprint. If a living neural culture is fundamentally driven by its nature to minimize uncertainty, then by carefully designing the sensory inputs and outputs, we can program it to learn complex tasks without needing the explicit reward engineering that traditional AI demands. We're harnessing the brain's natural epistemic tendency.
We've been discussing how biology's resilience stems from its plasticity and its inherent messiness, this massive parallelism of billions of components that are all slightly noisy. Let's formalize that messiness with the constrained disorder principle, or CDP.
If we connect this to the bigger picture, have you ever wondered why biological systems, which are built from inherently stochastic, noisy molecular components, are often so much more adaptable than our rigid, clean computer systems? The answer lies in how life has evolved not to eliminate noise, but to manage and actually utilize it. The CDP defines all systems by their inherent variability, the noise, and it gives us a platform for understanding when that noise goes wrong, causing a malfunction, or when it can be used to enhance functionality.
That fundamentally challenges the core scientific assumption that noise is just error, something to be filtered out, and that we only need the average, the mean measurement, to define a system.
It's a huge conceptual shift. According to the CDP, focusing only on the mean is insufficient because variability doesn't just describe the error, it characterizes the behavior of the system. The way the system fluctuates acts as a fundamental mechanism for improved performance, stability, and adaptability. The trick is the constrained part of the name. The system must maintain appropriate regulatory boundaries around that disorder.
So if the constraints are too tight, the system becomes rigid and brittle. If they're too loose, it descends into chaos. And when those boundaries fail, that's when we see pathology.
Precisely. Inappropriate brain and behavior variabilities, whether they become pathologically low, so too rigid, or pathologically high, too chaotic, are reliably associated with diseased states. These changes in variability are often the earliest signals of a malfunction in the regulatory boundaries. Managing the mess is critical for both health and successful synthetic engineering.
If variability itself is a key biomarker, where can we see this applied in, real-time life or death diagnostics?
The most accessible clinical example is heart rate variability, or HRV. HRV measures the subtle fluctuations in the time interval between consecutive heartbeats. We know that decreased HRV in preterm infants is a significant early biomarker, and it's strongly linked to negative neurodevelopmental outcomes. And the reason is that systemic inflammation, an early indicator of issues like neonatal sepsis, directly reduces HRV by constraining the autonomic nervous system's dynamic response.
So the lack of variability signals that the system is under stress and losing its adaptive range. How else is this measure used to guide complex medical decisions?
It's surprisingly versatile. Hemodynamic stability, tracked via HRV changes, is now used to guide anesthesia management, particularly in diabetic patients undergoing complex surgeries. Maintaining that dynamic stability managing the noise is key to better patient outcomes.
Have you ever wondered why this subtle fluctuation reflects the state of systems so far beyond the heart? I read that it even reflects kidney function.
It does because everything is interconnected via the autonomic nervous system. Patients with end-stage renal disease show significant measurable changes in their HRV dynamics. And the most compelling proof that the variability is functional is that these dynamics visibly improve after hemodialysis and substantially recover after a kidney transplant. The removal of the toxic burden restores the system's ability to fluctuate dynamically.
So beyond physiology, how are we harnessing randomness in advanced computational modeling and AI?
In AI and navigation, computational models designed for goal-directed navigation actively embrace variability. They predict the inherent errors and fluctuations in human path planning, which clarifies how uncertain, noisy sensory observations are integrated. By modeling the expected error, the system creates a more reliable cognitive map for route planning than if it just rigidly assumed perfect input.
And the most surprising application is how randomness drives the accuracy of those predictive biological clocks that estimate aging.
This is truly astonishing. DNA methylation clocks, which measure chemical tags on DNA to estimate age, rely on this variability. Studies using simulated data show that the underlying random process that governs these changes accounts for a massive proportion of the clock's accuracy, up to 75% for clocks like Horvath's clock, and sometimes even reaching 90%. The random process itself is not an artifact. It's a central driver of the measurement.
So if we fail to accurately model the noise inherent in biology, our predictive models will simply be inaccurate. This must mean conventional modeling techniques are running into trouble.
They are. Techniques like SINID, that's sparse identification of nonlinear dynamics, they struggle significantly when faced with realistic, sparse, and high levels of biological noise. To overcome this, AI researchers are developing frameworks using hybrid dynamical systems.
Okay, let's pause and simplify that part again. What does a hybrid dynamical system do differently here?
Essentially, the framework uses neural networks to approximate the unknown dynamics of the biological system. The neural network acts as a sophisticated, nonlinear filter. It's doing two jobs at once. It effectively denoises the sparse, noisy biological data while simultaneously learning the latent mechanisms that govern the system's behavior. This means we can correctly infer models and predictive rules even when the data quality is poor. It's the ultimate computational fulfillment of the CDP, using noise to improve understanding and function.
Let's transition from theory to some tangible breakthroughs. Synthetic biology is now designing cells to be miniature, smart drug factories that operate inside the human body.
This represents a massive leap in translational medicine. We are customizing mammalian cells. And these could be primary cells, immortalized lines, or even patient-derived stem cells that avoid immune rejection, to sense user-defined signals, process them using genetic circuits, and produce therapeutic agents on demand.
And how is the genetic program actually delivered into the cell to turn it into a factory?
We use standard delivery methods.
Mhm.
Now, chemical reagents, physical methods like electroporation or biological vectors, which are typically viruses. But for robust, stable, and precise long-term expression, we often harness high-precision genome editing tools like CRISPR-Cas9. That lets us integrate the entire synthetic construct into a specific, defined spot within the host genome, which ensures the cell doesn't lose the program when it divides.
Let's look at the fascinating open-loop systems where we provide an external trigger, a remote control, to turn the cell factory on. We have chemical controls, often tied to everyday compounds.
The examples are highly practical. Take C-star cells. They are engineered to sense coffee administration. A chimeric receptor is designed to dimerize when it's activated by a molecule in coffee, and that triggers a specific pathway, the JAK-STAT3 pathway, to express a therapeutic protein, HGLP1, used for type 2 diabetes management.
So my morning espresso becomes part of my precise, personalized therapy regimen that completely changes the concept of medication compliance and dose control.
Absolutely. The dose is directly proportional to how much coffee you drink. Then there are aroma cells, which express a receptor that senses volatile spearmint and, in response, produces a specific toxin. Or rapid cells, which are designed to release preformed proteins like insulin very quickly upon the addition of a specific small molecule.
And beyond chemistry, the physical cues are perhaps even more non-invasive and targeted: light, heat, ultrasound.
These physical cues offer superb spatial control. Opto-HEA cows contain the melanopsin receptor, the same one our eyes use to sense blue light, which triggers calcium influx upon illumination, leading to HGLP1 expression. A magnetothermal strategy uses magnetic nanoparticles delivered to the cell. When you expose them to a specific radio wave frequency, the nanoparticles heat up, stimulating an ion channel, which then causes insulin expression.
And for cancer, the physical cue can be applied externally to focus the drug production locally at the tumor site.
Precisely. For instance, you can achieve localized anti-cancer therapy by expressing a mechanosensitive ion channel on T cells. Low-frequency ultrasound stimulation applied externally at the tumor site triggers that channel, which then induces the expression of anti-CD19 CARs exactly where they're needed.
The most sophisticated applications combine multiple inputs into true logical processing. Tell us about the mesenchymal stem cells, the MSCs, engineered to detect and kill cancer using physical force as an input.
This is a complex, multi-component logic gate operating within a single cell. So non-immune MSCs are engineered with chimeric receptors that detect cancer biomarkers. But the critical component is an inhibitory phosphatase that sits near the receptor and constantly hampers the signaling pathway, keeping the cytotoxic drug expression off.
So the resting state is inhibition active, drug off.
Correct. But when the engineered MSC physically binds to a target cancer cell, the physical forces generated by that cell-to-cell contact are theorized to mechanically pull or push the inhibitory phosphatase away from the receptor. This removal of the inhibition triggers the synthetic expression unit to express and release a potent cytotoxic drug, killing the target cancer cell.
But wait, how confident are we that this physical force mechanism doesn't accidentally trigger a false positive on a healthy cell that just might be sticky? That sounds like a serious risk of off-target toxicity.
That's where the multi-input logic becomes essential. The system is designed to respond only if the receptor binds the specific cancer biomarker and the binding results in sufficient physical force. It's a very sophisticated AD gate. Biomarker detected. AD, physical force applied, equals kill signal ON. So researchers are focused on optimizing the receptor design to ensure high specificity and tuning that force sensitivity to minimize nonspecific activation.
That's truly advanced computation. And we can achieve complicated logic using purely DNA-based circuits too, right? Using hybridization chain reaction or HCR.
Yes, hybridization chain reaction uses short, complementary DNA strands that only start polymerizing and generating a long, detectable signal when they encounter a specific target DNA input. This mechanism can be used to generate complicated logic. For example, an integrated XOR&D circuit to autonomously evaluate multiple inputs within living cells.
And what's the ultimate application of putting an XOR-ND circuit inside a cell?
The goal is highly reliable cell recognition in cancer screening. The circuit is programmed to recognize the presence and absence of multiple specific microRNAs, which are often dysregulated in cancer. For example, this XOR&D circuit was successfully used to reliably distinguish specific cancer cell lines based on their unique miRNA profiles, showing a clear fluorescent signal only when the complex logical requirements were met.
The power of synthetic biology isn't limited to therapeutics. The same engineering principles can solve massive environmental issues, particularly the cleanup of heavy metal contamination through bioremediation.
Bioremediation using microorganisms to treat pollution is highly attractive because it's sustainable and potentially very efficient. However, natural microorganisms are often ineffective in heavy contamination scenarios. They evolved merely to survive in those environments, not to detoxify them quickly or efficiently enough for our standards. So synthetic biology enhances that microbial detoxification capacity through genetic reconstruction.
The first step in effective cleanup is precise detection. And engineered bacteria are incredible whole-cell biosensors for everything from arsenic to mercury and cadmium.
Let's focus on arsenic, which is a global drinking water contaminant. Researchers engineered an arsar-based biosensor. But to enhance its sensitivity, they introduced a Luxar-based positive feedback loop.
What does that positive feedback loop do functionally?
It creates an amplification system. When the bacteria detect arsenic, the system not only generates the output signal, but it also prompts the bacteria to produce more of the detection components. This self-amplification results in a much stronger response, allowing the system to detect arsenic concentrations as low as zero.1 micromolar.
And that's a crucial threshold, isn't it? Because it's below the World Health Organization's safety limit for drinking water.
Exactly. That feedback loop is the engineering magic that provides the necessary clinical sensitivity.
And what about detecting multiple pollutants at the same time without needing separate tests?
For that, they developed a dual output signal biosensor for mercury and cadmium. This system employs two different transcriptional factors. MER senses mercury and regulates the expression of green fluorescent protein, GFP. And at the same time, CADR senses cadmium and regulates the expression of M-Sherry, which is a red fluorescent protein.
So the cell lights up green if there's mercury, red if there's cadmium, and yellow if both are present.
Exactly. It provides a quantitative, visual assessment in a complex mixed metal environment. It can reliably assess concentrations up to 5 micromolar of mercury and 200 micromolar of cadmium.
Okay, so once detected, how do the engineered microbes perform the actual cleanup and remove the metals from the water?
The most common and effective mechanism used in synthetic bioremediation is sequestration. This involves engineering the bacteria to actively trap the heavy metals. This can happen in two primary ways, either directly on the cell surface via functional groups that chemically bind the metals, or by engineering the bacteria to secrete enhanced extracellular polymeric substances, or EPS.
These EPS are like sticky, high-efficiency nets.
Precisely. They are secreted proteins or polysaccharides that form a matrix outside the cell, designed to capture the metal ions and prevent their penetration into the cells, thus removing them from the surrounding water. It's a sustainable biological filtration system.
We've seen the extraordinary speed of this scientific advance, but for this revolution to move from the lab to reliable global deployment, the computational infrastructure needs to mature significantly. What are the major computational modeling hurdles facing this field today?
The hurdles are massive, and they're primarily centered around data. We face the challenge of multi-scale modeling, integrating vast, diverse data sets from genomics and proteomics all the way up to full physiological behavior. We also struggle with the inherent complexity of biological systems, scalability, the lack of standardization, and the persistent need for truly robust interdisciplinary collaboration.
This sounds like a classic challenge in a new scientific field. Everyone is collecting data, but they're using different names and protocols so the models can't communicate.
Standardization is paramount for reliable engineering. To promote model development, global communities like the Computational Modeling of Biological Systems Community of Special Interest formed back in 2016. Their whole goal is to organize collaboration and promote novel mathematical tools to handle this complexity.
When we focus specifically on neuro-AI and organoid intelligence, the data becomes immensely complex. You have time series, electrophysiology, high-resolution imaging, behavioral interactions.
For SBI and OI, standardized ontologies and metadata are non-negotiable for data sharing and reproducibility. An ontology is essentially a formalized naming system. We must rigorously document variables like the cell line, the organoid's exact age, the culture media components, the full training history of the experiment, and the precise electrode configuration. Without that detail, another lab just cannot replicate the finding.
And the volume of raw neural data must be staggering, requiring new ways to store and access it.
It is. This necessitates specialized, cloud-based infrastructure. Data repositories like DANDI, the Distributed Archives for Neurophysiology Data Integration, are vital. And major international efforts, including the NIH Brain Initiative and the Baltimore Declaration for OI, all emphasize that structured, shareable data is the only path forward. Without it, progress remains localized, slow, and non-cumulative.
Here's where it gets really interesting because these technological successes introduce profound ethical questions. We are designing systems that might, however primitively, be conscious. The development of brain organoids, which show spontaneous neural activity resembling early human development, raises major concerns about their potential moral status.
This is the critical, pressing conversation that needs to happen now. As organoids start showing capabilities like sustained memory formation or complex network oscillations, the ethical questions intensify. We often turn to frameworks like the Integrated Information Theory, or IIT, to help us navigate this.
How does IIT help us frame the consciousness debate in a biological system that doesn't have a full body?
Well, IIT suggests that consciousness, or morally relevant state, is related to a system's capacity for complex integrated information processing. It tries to measure a quantity called phi. If a system like an advanced organoid exhibits sophisticated integrated activity, it might possess what IIT defines as a primitive form of consciousness, demanding moral consideration regarding potential pain or suffering.
And the ethical challenge becomes particularly urgent when researchers shift from passive observation to actively creating learning systems, like those using closed-loop systems.
Exactly. Researchers are now implementing closed-loop systems that provide organoids with sensory feedback during learning experiments, simulating an environment. This dramatically increases the potential for creating a sentient system. Ethical guidance demands proactive monitoring protocols to detect signatures of sentience like integrated oscillations or complex memory formation, and requires multidisciplinary coordination among neuroscientists, ethicists, and policymakers before those experiments could proceed.
But what does this mean for our ethical obligations when we create systems driven by fundamental biological impulses, like seeking information to reduce uncertainty, as we discussed with active inference?
That's the core tension. If the system is intrinsically driven to explore and learn, it raises the bar for our responsibilities to ensure its environment is not one of persistent discomfort or stress. It forces us to move beyond simple technical assessment and grapple with fundamental philosophical questions about the limits of engineered life.
And then there's the issue of the original material, the human tissue donated for research.
Donor consent is a central and non-negotiable concern. If human organoids are derived from stem cell lines whose donors were not explicitly informed about the potential use of their cells in organoid intelligence research, that is an ethical failure. Donors must be informed that their cells may contribute to systems designed to acquire learning capabilities. And of course, safeguards must be strictly enforced to protect the privacy of the donor.
Moving beyond OI, what are the overarching governance principles for synthetic biology applications in biomedicine generally?
Governance is guided by three principles: human-centeredness, sustainability, and the critical principle of non-maleficence. And it's crucial to remember that non-maleficence is not a principle of zero harm, which is often impossible in clinical trials, but one of minimal, reasonable harm.
That distinction, reasonable harm, is vital for progress. Can you elaborate on what makes harm reasonable?
Harm is reasonable and ethically permissible if the trials are essential for testing safety and efficacy. The harm is proportional to the public benefit, and informed consent is rigorously obtained. For example, in human challenge trials for COVID-19 vaccines, participants knowingly accepted the risk of experiencing moderate symptoms. This harm was deemed reasonable because the resulting data was essential for the rapid development of a life-saving public health tool.
Finally, we have to ensure biosecurity, especially as techniques like those we discussed, fast, standardized cloning and high-precision integration via CRISPR make synthesizing and re-engineering life quicker than ever.
Global legal frameworks are reacting to this rapid bioengineering capability. We see frameworks like China's counterterrorism law, which mandates strict supervision and management of pathogens to prevent theft or illegal use. Biosecurity has to address not just physical risk containment, but also non-physical harms. We must critically evaluate the societal risks and potential for misuse inherent in granting humans the power to intentionally create and program new kinds of life.
This deep dive has shown us that the future of computation is quite literally alive. To help you integrate these three revolutionary areas, let's use a simple mnemonic we introduced earlier. LAV, the living future of computation.
L stands for logic. This is synthetic biology, where we program genetic circuits using DNA and proteins to perform precise functions. We discussed how standardized tools like SnapFast are overcoming the cloning bottleneck, allowing us to build complicated logic gates inside cells, like that XOR-ND circuit used for reliable cancer recognition.
I stands for intelligence. This is neuro-AI and organoid intelligence. These living neural systems use the inherent massive parallel computation of brain cells, following the principles of active inference. And AIF demonstrates that seeking information is a fundamental, self-driven behavior, but this emerging intelligence raises profound ethical questions about moral status and the need for new, rigorous donor consent protocols.
And V stands for variability. This is the constrained disorder principle. It teaches us that biological noise isn't merely error. It's a necessary mechanism for adaptability and a key diagnostic tool, as we saw with the clinical utility of heart rate variability. We're now developing advanced AI models like hybrid dynamical systems that actively embrace this noise to learn the true latent mechanisms of life.
The applications we explored span from using coffee to trigger on-demand therapeutic production and wirelessly activating T cells with ultrasound to fight tumors, to engineering bacteria that sense and sequester heavy metals below WHO safety limits. The power of programming life is expanding faster than we can regulate it.
The move from digital rigid architectures to plastic, cellular substrates requires a foundational shift in how we define what computation is and what it means to be alive. If biological systems trade silicon speed for characteristics like healing, adaptability, and potentially sentience, what ultimate applications genuinely demand a living computer over a traditional one? And where should we, as designers and as a society, draw the line on intentionally creating new forms of life with the capacity to learn and feel? That is the question we must carry forward.
Powerful questions to carry forward as this field matures. For joining us on this deep dive into the intersection of biology and computing, thank you for joining again.